Good afternoon, everybody. Certainly thanks to ARM for inviting us and allowing us to participate in these presentations. So as a company, Bluebird is really about hope, and it's about making hope a reality for the patients we desperately seek to serve. I will be making forward-looking statements. You saw that slide up there, so please reference our SEC filings. I just got off of a phone call with the entire company, all 450 people back at the company, and I don't know if you've seen, but we just recently announced that the data from our ALD program, more detailed data from the top-line data we gave a few months ago. It also is the subject of a New England Journal of Medicine article as well on this trial, where we showed 15 out of 17 boys having successful outcomes at the 2-year endpoint. More important than that data are the patients that underpin this. And the one story that stuck with me from the lead PI from the conversation back at the company was imagine the family around the bedside of a boy that ultimately has a potential death sentence due to the diagnosis. And they're all pushing a button to infuse the cells back, the patient's own cells back into that young boy with the hope over the next 2 years that their disease will stabilize. And the fact that the families universally break down in tears because they've been waiting for months now through this therapy, through the collection of the cells, ultimately the transduction of those cells, and they're coming to this moment where the cells of this patient are being infused back into the little boy's body. And this is your son. And really, really privileged today to stand up here and at least reference for the first time some of the detailed data behind that study. It is the anchor of the company originally. It was actually founded, Bluebird, on data from the CLD program, even though it gets lost sometimes in the shuffle because we have some other exciting programs too. But I wanted to really emphasize this, and it's the face of the boys right now that are in my head and in the head of the employees back at Bluebird right now. That is one program, but we have several programs. We have an anchor in the severe genetic disease program, so CALD is one of them. We're also treating patients with transfusion-dependent thalassemia as well as sickle cell disease. And then we have another arm to the company, another franchise as we call them, in immuno-oncology, where we have a CAR-T program in clinical development that some of you may know about with our partner Celgene. But it's the experience around these research experiments as well as clinical experiments that really informs the company going forward. And our learnings in sickle cell are a perfect example of that, how we've had to iterate constantly to improve the therapy and have the outcomes become better and better over time. And that will persist for the company. We will not rest on our laurels. We will leverage the tools that we have that we've accumulated, whether it be through licensing or acquisition. We'll leverage the experiences in both large market indications as well as later stage smaller indications, and certainly through the team that's been assembled back in Cambridge, and now we have folks in Europe as well as in Seattle. But it's all based on an ability to effectively and efficiently transduce cells, whether that be T cells or whether that be hematopoietic stem cells. And that foundational capability has resulted in a pipeline you see here with cerebral adrenoleukodystrophy as leading the way, along with transfusion-dependent thalassemia trial, with sickle cell not too far behind, and then our 2 clinical programs targeting BCMA with Celgene, and a pipeline behind that leverages both our gene editing capability as well as partnerships that we've established over time. And you can see we can't do this alone. We've got a tremendous number of partners that we work with. Obviously Celgene being one that's most pronounced, but many academic institutions and other industry players that are helping us along the way. So we painted this vision at JP Morgan not too long ago that we have a bold vision for the company to have at least 2 products on the market by 2022, 2 that are approaching commercialization, and a deep pipeline behind that, all with the goal of having transformative effects on patients. So these are not me-too products. These are truly transformative therapies. And the first 4 in clinical development have certainly shown that we're on our way, we believe. So how do we get there? So up here you see what we set out at the beginning of the year in terms of both data that we expect to be disclosing to the world, the execution that we expect during the year, and then other developments in terms of both additional clinical programs as well as advancing the pipeline. And you can see there are a number of checks here already. So we certainly gave you a first glimpse of the data in our multiple myeloma trial, the CRB-401 study. We certainly gave a sense for, at EHA, how we were progressing with our TDT program and a little bit on the sickle cell program. And then, as I mentioned today, as of today, we've given a more comprehensive update on our CALD program. We still have ASH ahead of us, where we will be providing an update across all of our programs except for the CALD program. We plan to start a 212 trial, which is the beta-0 trial for thalassemia, and then continue to inform the company as to our path toward regulatory filings, in particular around the TDT program and our CALD program. We've obviously started our 21-21-7 next generation BCMA program with Celgene, and we continue to advance the pipeline. But it's been an amazing year for us, and ASH is really going to be the culmination of that. So what I was going to do very quickly in the brief time that I have left is hit each of our programs and talk about kind of what's next. So CALD, here is the article that was published as of today on our HSC program for CALD. Very proud of this data. I'm not going to be able to get into the data in the short time that we have, but the trial itself is one that is an open-label, multi-center trial enrolling originally 17 boys. Those were the data that were just presented, that 15 of the 17 boys had reached the endpoint of MFD-free. Also, safety looked quite promising as well, as we had expected, lacking any GVHD. And this will form, along with the 8 patients referenced up above, which is an expansion to this trial, will form the basis of hopefully a submission to U.S. and European agencies for approval, but we need to get through these final 8 patients, which are enrolling right now. So really excited about the trajectory of that program, and more to come in terms of timing there. Multiple myeloma, I think people are familiar with the fact that we've had to date, as reported, a fairly manageable safety profile relative to other CAR T therapies as represented here, where we saw a limited amount of CRS and neurotoxicity, which does differentiate it a bit from other CAR T programs. And the rest of the AEs are associated with what you'd expect from the procedures these patients go through. From an efficacy standpoint, we saw quite compelling evidence of effect in the top 3 dose cohorts. The bottom dose cohort was not therapeutic, it was subtherapeutic, but the top 3 all had responders. So of those that we could measure response, we had 100% response in the top 3 cohorts at different dose levels. And as you know, we've now progressed to our expansion phase of this with a dose that ranges between the 2 middle doses, so 150 to 450 million cells. So at ASH, we will give an update on all of these patients, effectively 6 months more of follow-up. For our TDT program, these are the ongoing 4 trials where we are investigating our therapy for transfusion-dependent thalassemia. These are patients that are severe and dependent on transfusions to stay alive. Our 204 trial is the U.S. trial. 205 trial, which has sickle cell in it, is sickle cell patients as well, is the French trial. And then the North Star 2, HTB 207 trial, is the trial where we've enhanced the manufacturing processes, added 2 small molecules to the transduction process to allow for higher percentage of cells to be transduced, and ultimately leading to a higher vector copy number in these patients. The belief is that will lead to higher expression levels and ultimately a better outcome for these patients. Having said that, the 204 and 205 data that we've disclosed to date, we believe will form the basis, along with some supplemental data from the 207 trial with the new manufacturing process, for a filing in the EU initially, and then the full 207 trial will form the basis of a filing in the U.S. Again, this is for the non-beta 0 population. For the beta 0 population, this is the 212 trial I referenced before, which we hope to start and commence before the end of the year. So this is our registration strategy in the U.S., and as you can see, it's based on the completion of the 207 trial, which has these manufacturing improvements for the non-beta 0 population, and then the 212 trial for the beta 0 population. And in Europe, where we have an opportunity to have expedited process, seeking conditional approval based on the, again, 204 and 205 data, with some data from the 207 trial showing that the improved manufacturing process leads to higher benefit. We certainly have disclosed early data suggesting that we're on track as it relates to 207, delivering higher levels of functioning beta globin and therefore transfusion independence. And in fact, in the first patient, if you recall, we were able to get that one patient up to 13 plus grams per deciliter, which is normal ranges of hemoglobin production, functioning hemoglobin production. So that's incredibly exciting. We're getting even beyond just transfusion independence and possibly getting patients into normal ranges of functioning hemoglobin. In sickle cell disease, this is a part of the story that people are anxious to see at ASH. As you may recall, we went through a process in the company of assessing what optimization steps we needed to make to improve the outcomes. We had the initial patient that also was the subject of a New England Journal article, which showed that we can have a dramatic effect on patients, but then subsequent patients didn't do as well. So we went back to the drawing board and said, what did we learn from the two sets of patients? And this is represented here. It's a complex disease with a procedure that is complex. And so from the addressing the hypoxic inflamed bone marrow through pre-harvest transfusions to hopefully getting adequate myeloablation by optimizing the range of myeloablation, to making sure that we get enough cells and enough target cells out of the procedure, to making sure that the cells that we do collect have a high percentage that are transduced, meaning the gene therapy copy is in there. And then lastly, that we can optimally collect cells from these patients. And there are two different ways of doing that, either through bone marrow aspirate, which is historically the way we've done it, or through plerixafor. So at ASH, we plan to give an update on probably 2 or 3 patients with all of these changes except for the last one optimized. And our hope is that we will end up with similar to 1204, the 1204 patient outcomes. The other piece of data that we will be disclosing at ASH is the plerixafor arm of this trial, the 206 trial. That was intended to investigate the safety of using plerixafor to mobilize cells, similar to the way one does it for TDT, a fairly standard apheresis process. Today, the bone marrow aspirate process has generated a good cell count, but we think we can optimize that through plerixafor. So see early data on the safety aspects of that. Were we able to mobilize cells safely in those patients? Were we able to collect enough cells? And then ultimately, were we able to transduce those cells efficiently? We will not see at ASH yet the clinical data arising from those cells in vivo in the patients. That's more of a 2018 story. So in the moments that I have left, this is the story that we saw for sickle cell. This is looking at drug product vector copy number versus in vivo peripheral vector copy number. The first patient that I referenced, the 1204 patient, had a very consistent and stable DPVCN to peripheral. The ones in the blue were the ones that created that aha moment that we need to learn more and advance the manufacturing processes. Well, you can see in the dotted line patients, these are all patients that have been optimized for 2 components, 1 for myeloablation and 2 for transfusions, but they don't yet have the other 2 components, yield and ultimately the improved transduction process. So that's what's to come at ASH. But you can see that we believe that we made some improvement. Those patients are having a higher vector copy number, and those same patients, also shown in the dotted line, are having a higher T87Q level, meaning functioning globin. So we hope to show that at ASH. These are the things that you'll see at ASH for 206 in terms of what's been optimized. And then lastly, it's really all about the people, the 450 people back at Bluebird, and the excitement around the prospect of the company moving forward, not only with a couple toward hopefully filing and registration, but also moving into phase 3 with the other 2 programs. So an incredible amount of data coming out at ASH, so stay tuned. Thank you very much.